Hello everyone. I'm Charmaine Demanuele. I'm a director of Biostatistics at Pfizer, and I lead statistics for digital medicine and translational imaging group. And I'm delighted to be here with you today, together with my colleague, Tomas Adamushek, who will co-direct the panel with me. So I'd like to start by thanking the organizers for putting this fabulous meeting together and getting us together to discuss this really important topic. And thank you for the opportunity to be here today. In this panel, we'll be talking about leveraging multimodal sensors to give us more precise endpoints. So we all know that digital health technologies enable us to simultaneously measure so many things about our patients. We can capture physical activity via accelerometers and gyroscopes. We can look at physiology with heart rate and temperature. We can look at cognition and emotion with touch screens, microphones, and cameras. And also we can get information about the environment based on light, sound, and location. So the challenge really is how to combine these different signal modalities to better phenotype our patients, characterize their symptoms, and measure their change over time. So we are lucky today to have these highly talented panelists with us to talk about current examples of how multimodal sensor integration is being used both in large-scale observational studies and in clinical trials. We will also talk about statistical approaches for this type of data, considerations for technology selection, and how to capture the patient perspective to derive clinically meaningful endpoints. So we'll start with a round of introduction. So Tomasz, you can start by introducing yourself, and then we'll go in order.
Hi, I'm Tomasz Adamushek. I lead the data science team in the digital medicine at Pfizer, and it's a great pleasure to be here with everyone. Thank you.
Vadim?
Hi, everyone. I'm Vadim Zipunikov, an associate professor at the Department of Biostatistics at Johns Hopkins School of Public Health, and I work on methods development and understanding signals and data from wearables and smartphones.
Thank you. Giorgio?
Hi, everybody. My name is Giorgio Guerra. I'm the Director of Artificial Intelligence at the Scripps Research Translational Institute, and I lead the team in data science in particular.
Dmitry? You're muted, Dmitry.
Hi, Dmitry Volson. I'm leading analytics team at Takeda, working on developing new methods and quantifying sensor digital data across therapeutic areas. Pleasure to be here.
Emily?
Hi, I'm Emily Cerciello. I'm Associate Director of Digital Health and Engagement at the Crohn's and Colitis Foundation, where I facilitate patient engagement in developing new technologies for research.
And Matt?
Good morning, everyone. My name is Matt Wilkes. I'm an intensive care physician and physiologist based in Scotland, and I'm the head of research at Current Health, which is a wearable device and care integration platform.
Wonderful. So thank you for the introductions. So Giorgio, we'll start with you. Can you walk us through an example of how you've used multimodal sensor integration and some of the lessons that you've learned in the process?
Absolutely. Thank you for the question, Charmaine. So let's talk about one of our study with multimodal and multi-sensor data, which is DETECT. DETECT is an app-based research platform that it has been designed in March 2020 as a response really for the COVID-19 pandemic. The idea of this study is basically to recruit. We have been able to recruit about 40,000 participants, and that the participants donate their data as part of the study. They can donate sensor data on one side, but also self-reported symptoms, self-reported test results, and they can connect their electronic health records. So we have really a multimodal platform. And what we want to do, well, the first goal was really to be able to detect COVID-19. We've been the first group that was able to use sensor data from wearables for the detection of COVID-19 on a publication in October 2020 in Nature Medicine. So there are several challenges in using this data. The one that I've been most excited about was to be able to integrate data from very different sensors. We've been using some participants share data from Apple Watch, some other from Fitbit, for example. And this different sensor have a different way to interpret even some very simple data like the resting heart rate. The values are slightly different between different sensors. So there was a big question here on how we approach them, having multiple sensors from different people. And there was another issue in each of these sensors besides resting heart rate provides also, for example, sleep or activity data, how we bring all of them together. Just in a short answer for that was for us to focus on individual data. So instead of trying to find something that works for everybody, we really focused on the individual, and we look at the individual to figure out what is changing over this multiple dimensions of data. What is changing for the individual? We figured out what is normal changes for the individual and what changes that may be due to an infection like COVID-19, and in the future, other different type of viral illnesses.
Thank you. That's very interesting. Dimitri, going over to Dimitri, could you tell us a little bit more about strategies of employing multi-sensor approaches in clinical trials?
Sure, thank you. I just want to put everyone on the same plate in terms of clinical trial as opposed to maybe larger observational studies. So just to remind everyone that we have phase 0 to phase 4 trials, and of course in phase 0 we are looking at the things like bioavailability of pharmacokinetic properties, and in case of digital trial that would be potentially testing new digital modalities in population of interest to understand how they quantify patient behavior against clinical controls. So in phase 1 trial, we are testing drugs in healthy volunteers. These are usually those single dose or ascending dose trials, and the goal of them is a safety check. However, we're also using often devices and multiple modalities into probing how potentially we see some signs of efficacy or safety concerns in the context of healthy volunteers. So for phase 2, these trials are targeted towards testing efficacy and side effects in a larger population, more sizable set of subjects in the population of interest. And of course, here the goal is to use devices across multiple modalities to establish secondary or exploratory endpoints that would be testing the efficacy and potentially side effects. So in phase 3 larger trials, we are looking for therapeutic effect, and beyond efficacy and safety, we're also trying to establish effectiveness, therapeutic effect of the drug. This is the primary interest for drug development. So a lot of efforts in the prior trials are typically targeted towards being ready for phase 3. And finally, last but not least, we have also phase 4, which is retrospective post-marketing monitoring of patient population. And here the goal could be potentially looking at how we can better understand how drug works in a larger patient pool and potentially look into extending patient population. So within this framework from phase 0 to phase 4, I just want to mention few important aspects. So one is, several people already mentioned that the holistic view of integrating multiple sensor modalities. So this is the primary interest of putting multiple sensors, is creating complementary view on physiology, behavior, cognition, and multiple components, understand them in helping each other to understand the holistic view. So the second point I want to make is the sensors are, even good sensors are typically not measuring exactly what we want. And so there could be multiple reasons. The concept of interest could be fuzzy, or signal could be measured, but with a good sensor, but it creates a burden. So we need to bring multiple sensors that would be easier to use and combining them to approximate somewhat better sensor. We also sometimes don't know exactly what to measure, and that creates a need to bring multiple sensors to better understand the relationship to population phenotype, as well to efficacy of the drug in rescuing these phenotypes. So the other aspect that I want to mention is that when planning all these studies and going through all these phases is really helpful at the very beginning to plan for life cycle management, plan from concept of interest, what kind of devices should be used for this concept of interest, but then how we will capture safety, efficacy, and how we will convert all these learnings throughout this phases of trials, 0 to 3, how we'll convert it to potentially a product, or how we'll understand how drug works in post-marketing world. So the other item, which I think is important to mention, especially regarding phase 0 to phase 2, and this is to contrast with the population larger studies, is that these are typically very small studies. So we cannot really test and combine our sensors and test all the models and validate them in the same study. So it's of utmost importance to bring external data from larger historical studies or public databases. These sensors may be dispersed across multiple studies to build the models and then tune them for your patient population and validate them within your smaller studies. So I think this is one of the keys and key differences with larger population type of work. So in our work, I just wanted to mention that we are largely in one of our major focuses is on narcolepsy, and this is where narcolepsy phenotype is related to sleep. So the key cardinal features of narcolepsy: excessive daytime sleepiness, disturbed nighttime sleep, and cataplexy. And the key challenges in our work in working with this type of population is, of course, the fact that the diagnosis of narcolepsy is extremely delayed. So it takes 5 to 15 years to diagnose the subjects, and that creates sort of opportunity for us to bring in digital technologies to enable earlier detection. So the in course of the trials, the cardinal features such as excessive daytime sleepiness and disturbed nighttime sleep are usually measured in the clinic, and a lot of variability and limitations of measuring quality of wakefulness and quality of sleep through in-clinic studies is another area where combining multiple sensors and measuring things at home continuously could bring a lot of value. And this is what we are focusing on. Oftentimes, combining sensors, even simple sensors, brings a lot of opportunities. And as an example, accelerometry and heart rate combining together could enable large set of both safety as well as efficacy endpoints, and this is something we are actively working on.
So thank you, Dmitry, for this really comprehensive description of how you're using multimodal sensors in clinical trials. So as you and Giorgio both mentioned, fusing data from multiple sensors brings a whole new set of challenges for analysis and statistical modeling of these endpoints, right? So Vadim, can you describe some of the statistical challenges and tools you've been used to mitigate them?
Absolutely. So in the next, I guess, 5 or so minutes, I will try to cover 4 different things. First is the temporal aspect of the data, because this data is collected over 24 hours. Also distributional aspect, which is quite important. I'll tell you specifically what I mean by that. Also, how to define baseline and how to understand that there is a change in this subject-specific baseline. In, as you said, fusion, right? But before going to the fusion directly, we want to understand each modality separately. So this is why let's start with the temporal aspect, right? Like you have this multiple devices, or you might use a smartphone for reporting something, and then this data is being collected over 24 hours. So understanding how at the subject level, a specific variable, for example, heart rate changes over 24 hours is quite important. And in statistics, there are so-called functional data analysis and time series analysis tools, which allows you to do that. And within the functional data analysis, you could think about, okay, you might have subjects, one with high disability and now one with low disability, and then the difference will appear, say, in the second half of the day when there is an emergence of the fatigue. Right. So in that case, this is where functional data analysis would allow you to detect this difference. When you specifically keep track of the time of the day and you model this time of the day specifically in your model. Then another important thing which can be captured in the using functional data analysis tools is the technically speaking, phase and amplitude, right? So you could imagine that two subjects or two participants have exactly the same daily routine, but one subject wakes up 2 hours later, right? But then there is a shift in 2 hours, but essentially the days are exactly the same. So if you just look at the time of the day, then you would see some differences, but in reality, the major difference is in the phase, right? So this is where you could use phase and amplitude separation within functional data tools. Another important aspect you can capture is a data quite often sparse, right? Like, for example, if you're modeling gait, these walking bouts happen sporadically from the day and very sparse, right? So how to, from one side, capture difference, the diurnal difference, that the way you walk in the morning and the way you walk in the evening might be quite different. And also you have this just bouts or episodes of walking. And this is where sparse functional tools allows you to actually deal with this. Another interesting aspect is that quite often we have temporal binary events, right? So for example, if you have a cardiac patch and it tries to detect, say some irregularities in the heartbeats, then you might have episodes of the atrial fibrillation, right? So this is where you could also use these tools and see if there is a higher likelihood and a higher probability of having those events in the evening or during nighttime, which is quite often might be the case. And this is where you could actually, by incorporating time of the day, you might be create a more powerful endpoint and more powerful summary. So just to summarize this piece, you could use this temporal and diurnal profiles both as the outcomes or as the predictors in your models. And this is where functional data tools are quite efficient. Then an important thing, I think, is the how to define baseline and what is the difference in the baseline, right? If you have multiple sensors, you just can go into a specific sensor. So Dmitry, for example, mentioned having both accelerometer and the heart rate, right? And like if the person is doing some physical activity, then we could see elevation in the heart rate and vice versa, right? Or if the person, for example, taking a beta blocker, right? We might not see at the subject level this linkage between heart rate and physical activity, because the beta blockers keep it at bay, the heart rate level. So we would like to understand all this. So even this relationship or correlation between one sensor and another sensor might be baseline, right? And the change in this in this correlation might be an important parameter, or might be actually chosen as an endpoint. So more broadly, we try to understand, when you have multiple sensors, we're trying to understand this joint dependence between domains you measure with these multimodal sensors, right? And the change in the strength of the correlation might be actually quite informative, right? So because people sometimes kind of turn on some compensatory mechanism, and we might not see it at the beginning, and we might see it emerging later on. So that might be quite an important advantage of using multimodal settings, multimodal toolkits. And also coming up with the composite endpoints, I think it's a big advantage of using multimodal modeling. But of course, it's pretty tricky. So I think people thinking about this, but how exactly to come up with this composite endpoint, I think is still a work in progress. But also that this is also tells us that understanding each specific modality and understanding its value, this is what we should focus on right now. And it looks like my time is almost up, so I'm gonna stop here.
Thank you. Very interesting insight. Matt, over to you. A question: in your experience, what's the most important features? What should drive the technology selection for multimodal sensors?
Thanks, Tomasz, and thanks everyone for some very interesting comments so far. I think when we're looking at technology to pick these endpoints, there's the first thing that Dimitri said, which is important to have real clarity on the experimental question. You have to know exactly what you're trying to measure. And that's not just in the case of particular parameters like heart rate or stats, but also, like Vadim was saying, the interaction between those and how that's going to play out in your endpoint. I was actually very interested in what Giorgio was saying, because at Current Health, we take a slightly different approach. So where Giorgio is combining large numbers of different devices, we have a single wearable device with integrated sensors. They measure oxygen levels, so oxygen saturation, heart rate, respiratory rate, motion, and temperature, all from a single wearable. And so the way we approach what technology is appropriate is we start by saying, what's the question? We make sure that we can capture those with our sensors. But one of the things that we think very hard about is validation. And there's validation in terms of things like FDA clearance. Do you measure what you say you're going to measure? And not all wearables kind of are able to do that. But more interesting to us is how is it going to be used? Has it been validated in the setting in which it's being used? So for us, just knowing that a sensor measures what it says it does is not sufficient. What we need to know is that it's going to work in that patient's house, when they're moving around, when they're doing their activities of daily living, and that it's going to work across its full operating range. And so it's assuring yourselves of those things through things like human factors engineering that give yourself confidence that the patient is going to collect what you would like them to. The other thing that we come across a lot that's important is who's going to be watching this. So your technology selection is going to be partly dependent on how you're actually going to be monitoring the patient. Are you going to be monitoring the patient in real time? Are you going to be collecting the data for research after a particular period of time? And it's these things and making sure that you have a very good way of data triage and data display that I think are just as important as picking something that measures oxygen saturation in the way you like. And all of this comes down to making sure that you're gathering the data you need to answer the question in a way that's validated, in a way that's going to work for the patient, but also in a way that is convenient for the patient. And so one of our biggest focuses at Current Health is adherence. And we make sure that our wearable is usable by people from all backgrounds and of all educational levels. We're very conscious of things like the digital divide, so making sure that people have access to things like internet and cellular. We build some of that into our products because I think having these tools is wonderful, but unless somebody is using them, it's not going to enable us to achieve the endpoints we would like. I think the final bits for us is that as long as you've got something that is working well and meets your needs, the next thing is to be constantly refining. And so we invest a lot in post-market surveillance and in working out ways to iterate our product based on how our pharma partners have found it for particular trials. And some of the things that have come out of that are things like making sure that data transfer is slick, that data handling is easy for those trying to work out the trial endpoints, that any algorithms we use are highly explanatory and very transparent. And then really just working with providers to make sure that when the trial is wrapped up, we have been able to take all of the lessons learned from that and apply it to the next.
So Matt, thank you so much. This was like really interesting, and it ties very, it basically is a really nice introduction to what I want to ask Emily, which is the patient perspective, right? I really want to understand from Emily, what should we keep in mind as we're collecting and analyzing this multimodal data from the patient's point of view?
Thanks, Charmaine. I'd like to make a couple points on design, data collection, and data sharing when it comes to the patient perspective. So starting with design, the importance of the visual appeal, comfort, size, and form factor will somewhat depend on the technology, whether it's worn, for how long, the severity of disease, and individual motivation. But in general, every step or click to activate, adjust the device, or provide data matters, and Matt is probably very well versed in this. But depending on the disease we are studying, factors such as stigma and attitudes towards using a piece of technology, the ease of use and reliability can be barriers to acceptance. For example, for patients with symptoms like dementia. So when designing a sensor device, the importance of understanding the specific needs and attitudes of the target patient group cannot be understated. Next, on data collection, I want to first mention informed consent. In research, informed consent is required, except sometimes when it poses minimal risk to the participant. And for large-scale ambient multi-sensor studies, this may be infeasible due to de-identification techniques and the multimodal nature of the data, but I don't have an answer to these ethical challenges, but it is important for the field to think through them. But in most contexts, we would ideally have a comprehensive informed consent detailing how and what data will be used by whom and for how long. Second, on data collection, individual baseline is really important, as Vadim mentioned. Understanding what is meaningful to patients means understanding what a meaningful change is to an individual patient or to a subgroup of patients. Authentic engagement of diverse patients in the development and selection of endpoints, especially regarding understanding meaningful changes in disease progression that can be measured by multimodal sensors. This may or may not differ from what changes that clinical teams see are as important, but it's really important to engage patients in this process. Additionally, intersections of behavior and other context information may be helpful to derive interesting conclusions. PROs are an added dimension of the multimodality aspect that we haven't touched on yet. I don't believe we're yet in a position to collect all of the data relevant to the patient and the clinician using passive techniques, so it is important to talk about PROs here. And this is related to the last point I want to make around data sharing and interpretation. For participant engagement, it's important to align the why of the patient with the product features. As much as possible, try to draw a direct line between their participation and their benefit, or a benefit to patients like them, especially when PROs require active work in addition to passive data collection. The PRO intersection provides an opportunity to provide value back to the participant, which might look like providing info about their health status, some idea of how data is being used by researchers, or a moment of delight to improve engagement and create a feedback loop. Careful consideration must be paid to interpretation within the multimodal setup. If sharing data back with participants, it's key to describe how one mode of sensing data alone might not give the full context, and incorrect interpretations on the part of the patient could be suboptimal or even harmful. So it's up to the research team to make the proper use of any data shared back to the patient entirely clear.
Excellent, Emily. Those are really good points, because when we think of multimodal, right, we think about different sensors and how we can link different sensors, but there's also other concepts of multimodal data that we bring in, such as you mentioned, the PRO, and that's equally important. And we need to really understand how to best capture it, model it, and interpret it. So while we're in this topic, can you talk about, Emily, one disease area that you feel could benefit greatly from multimodal sensor integration and maybe has not been really leveraged to date?
Absolutely, that's a great question. So I really think that diseases that cycle through periods of remission and relapse have been largely untouched. For example, many autoimmune diseases where patients are symptom-free for a period of time and then have flares of symptoms or exacerbations. There's been a bit of work related to rheumatoid arthritis and pain management, but new multi-sensor digital health technologies could be deployed more broadly in autoimmune to understand environmental triggers or physiological predictors of relapse and flare, or to predict and measure responses to treatment.
Thank you. And Vadim, maybe one last question to you. Where do you think we should be investing more when we think about future methodology work?
Yeah, that's a great question. Again, I think I just working in this field for like last 10 years. I think there is still a lot room for improvement and a lot of room for understanding what exactly is important for patient and what's informative about the status of the patient, right? And then, so I think that this is why attracting talent in this field, I think it's really important. So that's.
Yeah, absolutely, absolutely. And what are the advances that you see in technology that could help us basically accelerate the field forward?
I think there's a few things to move things forward. I think the regulatory climate is important. I think we need to really clarify what counts as a validated sensor, and also get acceptance from the FDA of wearable and remote measurements and purely digital endpoints as being acceptable currency in a trial. And if there's additional validation required to do that, then understand what that is. I think that's essential. In terms of the sensors themselves, I think they're pretty good. I think that there are areas where certainly we can do more work. Things around, I think continuous blood pressure monitoring is one of those things that has always been promised and has never quite appeared. And I still really don't have faith in what's out there. I think making sensors smaller and also combining them in single devices just to reduce patient burden, I think is a really good step towards adherence. And then finally, I think it's going to come down to how we use not just the sensor values themselves, but their interplay, the entropy and the information transfer between those sensors to get these kind of composite or surrogate endpoints of some of the more tricky or elusive things that we're looking for, a bit like Dimitri described.
Wonderful. So we have a question actually from Marta Karas. And she says, related to individual baseline for better individuals assessment, are there any means to access relevant data from before an individual enters the trial or study, example, long-term mobile phone logs?
Yeah.
And this question can go to Giorgio. Yeah, I think that would be a really good.
I can start with that. So thank you, Marta, for this interesting question. What we do, at least in our study, so we use sensors that the participant is already wearing, and this allows us actually to ask the participant if they want to share data also before the moment in which they really consent. So we ask for access to previous data. Companies like any company is really tracking, of course, all the wearable data that has been collected. So if the participant allows, we can go back. This is really important because this allows us to build a baseline for the participant. This allows us to understand how the participant is using the sensor and how the participant is really behaving. We've been talking a little bit about the different activity over the day, or how the heart rate may change. And this is really important also to understand how it changes not only over the day, but over weeks and months. And understanding what is normal and what is, let's say, outside of the normal range for a specific participant. And we are really different. Before starting our study, we have looked at about 200,000 individuals to really understand a signal as simple as resting heart rate. What does it mean? How does it change over time? And if I can make one last comment here, I think this is a really unique opportunity. We're talking about how accurate they are, the sensor, and how how we can measure different things. But one really important aspect is that this is a longitudinal and passive way to monitor an individual. We have never had this opportunity before. This is an opportunity to track an individual potentially over a lifespan and really understanding what's working with the individual and when something is changing for a specific individual. So, and of course, return this information to the individual at the end. Thank you.
Wonderful. So there are no more questions for now. So Dimitri, any maybe further considerations about multimodality in clinical trials or insights that you can share from your work? I know we cut you short, so I know you have a lot to say.
Oh, no worries. So I just wanted to add to Marta's question, actually, as it relates to clinical trials specifically. So I think one important aspect here is that we are, let's say, in phase 1, phase 2 trials, we are reporting baseline adjusted, placebo-corrected endpoints, whatever they are. And when we have 10 patients in clinical, in a placebo arm, then it's extremely important that these patients would represent the appropriate population. So let's say if you have control case versus control, then the clinical controls, as an example with the sleep, should exhibit normal sleep. So that means that there is a huge value in pre-screening subjects using sensors and selecting the right patient population. Because of the tremendous amount of effort and price that goes into the clinical trial, and it's often really painful to see 1 or 2 clear outliers. And using pre-screening, we were able to identify, say, subjects who shouldn't be perhaps admitted to the clinical trial, and that will save a lot of cost for in terms of drug development.
To pick up on Dimitri there, I think that's really interesting, because one of the things that we find when we deploy current health clinically, which we do in a number of health settings in the States, is just the sheer number of incidental findings, particularly overnight. Like the number of patients who have some element of nocturnal hypoventilation or sleep apnea, which, you say, if you hadn't pre-screened them, I would imagine could really mess up one of your phase 1 or phase 2 trials. And the broader point that I think Giorgio was making is, with my other hat as a physiologist, one of the things that I always struggle with is inter and intra-individual variability when designing studies. And it does strike me that continuous monitoring has huge potential here for us to understand exactly what a normal time course is for an individual, and then to truly make sure they are a control.
Yeah, absolutely. We have another question, which is, I think maybe directed to Dimitri and also to Giorgio. Are you generally using off-the-shelf sensors or developing custom sensors for multi-sensor monitoring?
Giorgio, you want to start first?
I can start, of course. Yeah, thank you. Well, what we use off-the-shelf sensors, at least we do not develop new sensor at Scripps Research. We are using any new type of sensor that is coming out. One advantage, there are advantages on both sides, of course. One advantage of using off-the-shelf sensors is that the individuals already have them, to some of them, and so they can participate in the study without the need of distributing sensors. And this allows us to really scale up much quickly and to reach many more individuals. And I can connect it with what Matt just said, but I think Dimitri maybe have another point to make.
Well, I think when we are saying sensors, I would assume that it's a combination of hardware algorithms as well as the practical ways of implementing them in a clinical trial. So we are using a mix of off-the-shelf things for first and simple looks, but because specifically in narcolepsy, the patient sleep is very different from other patient populations. We have to design improved algorithms because in most of the cases off-the-shelf simple sensors just don't cut it. But in other cases where we want to pre-screen subjects to make sure that the clinical controls have normal sleep, we don't. We can use something as simple as actigraphy and just look at the charts.
Can I just add a quick point on the off-the-shelf versus custom? Just an opportunity to make sure we recognize that patient representativity is really important, representing the underlying patient population. So I always have a little bit of concern when we just take sensors that are already out in the wild, because it could be a self-selecting patient population. So custom deployment of custom sensors can really make sure that we're hitting diversity in terms of enrollment.
I think that's a really interesting point. And this really alludes, I think, to the two approaches you can take, which is if you need very, very large numbers of people in the sort of tens to hundreds of thousands where you don't want to do the distribution, then really at the moment, Giorgio's approach is the only option. If you need to have a device that is transparent, where you can retrospectively audit the signals and where you need to be assured that it can work in patient populations of all backgrounds and with all abilities, then I think at the moment a custom solution is probably best.